Robert is a professor at the University of Minnesota. As he's a Denali Fellow. He works for Denali Pharmaceuticals as well. So, Robert, great to have you here, and looking forward for your presentation.
Great. Can you all hear me okay?
Yes.
Yes.
Okay. So, yes, thanks, Joe. So I'm full-time at Denali Therapeutics, which is a biotech in South San Francisco here. We focus on neurodegenerative diseases. And I thought I'd start by giving you a bit of an overview of, the field that I'm in, which is the larger sort of drug delivery to the brain field, CNS drug delivery. And if you think about where we are today with large molecules, so I really focus on large molecules. I'll keep most of my comments to that. Brain delivery, there are 9 approvals in the United States, FDA approvals, and it's kind of almost an even mix between invasive. So this is intraparenchymal injection. There was actually the first ever approval earlier, just like a month ago, a few weeks ago, of a gene therapy approach with a convection enhanced delivery into the striatum. And then the CSF administration approach is intrathecal and intraventricular. Over on the non-invasive side, there's a lot of interesting work, and I'll mention the sort of like biological mechanisms, exploitation of receptors and transporters at the blood-brain barrier to cross the BBB, get into the brain. This is what I am currently focused on at Denali. But there are a lot of other interesting avenues also that are being pursued, both experimentally, and then there are a few approvals. One experimental approach is focused ultrasound, which has also been paired with intranasal administration. It's an interesting combination there. But then I include here, and this is actually from a review a couple of years ago by Jörg Terstappen in Nature Reviews Drug Discovery, and intranasal drug delivery is here as well. And that's, of course, the concept here, nose to brain, bypassing the blood-brain barrier. So it's something that I've been in for a long time. You can look at this book here that was published a couple of years ago to get more of an overview. So now I want to basically transition and spend the rest of my talk on nasal drug delivery. So thank you, Joe. You already mentioned comparative aspects of nasal passages. And as you also mentioned, a lot of the work that's been done in this field's been done on rodents and preclinical species, non-human primates, macaques, dogs, and rabbits. And so I just wanted to mention very quickly, that it's very important to keep in mind when we're thinking about cross-species translation, how do experiments in rodents translate to what's being proposed or being done in human beings, is to keep in mind the differences. And so Joe already mentioned that there's a real difference in the relative proportion of the area or the volume of the nasal passage that's devoted to the sense of smell. So it's about 50% in the rodent, and it's maybe around 10% in the primate, less than 10% in human. Another one that I would suggest is important is just in thinking about the architecture of the nasal passage. And so show this over on the right here. If you look at there's this comma-shaped architecture of the turbinates in the primate, and you go and you look at this turbinate structure in the rat, for example, shown over here, and it's a very elaborate scrolling pattern, and I'll show you some pictures of that in a moment. So the takeaways are that the turbinate complexity is much greater in the rodent than it is in the primate. Higher olfactory surface area. And then something that I don't really have time to go into, but you can look at this paper that my academic group published about 10 years ago now. And I think there are also differences in the vascularity of the different parts of the nasal passage. So I'm going to focus really mostly on talking about the olfactory versus the nasal respiratory mucosa for the rest of my talk. So I've been in this area for a long time, and I think safe to say I've contributed in each of these 3 areas, understanding the anatomical pathways into the brain, the anatomical pathways for drug delivery, the olfactory pathway and the trigeminal pathway, and then what happens once you're in the brain for further distribution, widespread distribution to different structures. And I have proposed that involves perivascular biodistribution. So there are a lot of papers that have been published on this. And so my first paper in this area was almost 30 years ago now. And I'll sort of give you a little bit of an overview, and how where we are today, but beginning with where we originally started. So I was a PhD student, a graduate student with Bill Fry, some of you may know, at the University of Minnesota a long time ago, early 90s. I joined Bill's lab initially in 1990, and I started to work on this project, which was basically the idea maybe we could deliver neurotrophic factors into the brain along the olfactory nerves. And I like to sort of remind people of this history because it's important that the Bill's lab was an Alzheimer's research laboratory. We had a brain bank, so it was an AD focus. And the idea for this nasal route of drug delivery to the brain, at least in Bill's lab with him and I, came about from knowledge of the pathology of neurodegenerative diseases. And really, what was in the Alzheimer's field, almost an olfactory vector hypothesis for the causation of Alzheimer's disease, namely that a bacterial pathogen or a virus or something in the environment, like aluminosilicates, might gain entry to the brain. And a lot of neuropath work in the past, sort of trying to sleuth out, well, where does Alzheimer's disease start? And you look at sort of like the Braak staging, if you know anything about this. These olfactory areas of the brain are involved very, very early on in the disease. And that's true in both Alzheimer's disease as well as in Parkinson's disease. And so Heiko Braak is really known for this, the Braak staging in both Alzheimer's and Parkinson's. And so you have the pathology starting in these olfactory areas very early on. And then if you sensitively test for the sense of smell, like work from Richard Doty at the Monell Chemical Senses Center at the University of Pennsylvania, I think it is, you can actually detect olfactory deficits in Alzheimer's and Parkinson's before the classical symptoms, memory loss, motor symptoms of the disease. So that was essentially, this important, very important background, why we started to look at this initially. Then you have the beautiful sort of anatomy of the olfactory bulb, the olfactory system. And the important thing to mention here is that olfactory neurons it's really the only example of direct extension of the central nervous system in a distal epithelium. So the dendrites of these cells, where the odorant receptors are, I'll just mention after the first talk, that Linda Buck and Richard Axel won the Nobel Prize in 2004 for their description of this superfamily of G protein coupled odorant receptors. That was at Columbia. That these cilia with the odorant receptors are in contact with the external environment. And so if you get into these neurons or components associated with these neurons, the olfactory sensory neurons, you basically hop one synapse and you're into these areas that are then implicated in the neuropathology of these diseases. So that's really, that was the background. So the olfactory mucosa, the anatomy and physiology, I don't think I have to really tell any of you on the call here too much about this. It's a pseudostratified columnar epithelium. There are three principal cell types: the olfactory neurons, I've in my cartoon in yellow, these sustentacular or supporting cells in blue, and then these underlying basal cells, which can undergo mitosis and actually regenerate. They produce a lifelong supply of these sustentacular and olfactory neurons. Now, if you get by this, so olfactory epithelium is a barrier like all epithelia. It has tight junctions. There's a lot that I could say about all of this, so I'm going to go pretty quickly through it. If you get past this barrier and get to the lamina propria, or what's called the submucosa, then there are three principal fates that I might think of. One is you can get absorption into the bloodstream. Another would be absorption into the lymphatics, and the lymphatic drainage of the olfactory mucosa, at least, is to the deep cervical lymph nodes in the neck. And the third is to travel along these components of the nerves, the phylloolfactoria, the olfactory nerves, into the brain. And so essentially you can simplistically think of that. And so obviously these olfactory nerves then go to the olfactory bulb. So as I mentioned, about 30 years ago, more than 30 years ago, I started to do work on this with Bill Fry. And we published this paper in 1995 where we were administering tracers. So this is wheat germ agglutinin horseradish peroxidase. It's a 62 kilodalton protein conjugate. And we looked at the intranasal, sort of the ultimate fate of this molecule when we administered it intranasally in rats. We compared it to horseradish peroxidase, the part of the tracer that allows us to visualize it using a reaction with diaminobenzidine. So it's you can look at it. It's a visual using the microscope, or using as in this case we used an ELISA to quantify the concentration. But we use both microscopy and then biochemical methods to quantify that. And what we saw is, and the experiment was quite simple. We wanted to know, what's the capacity of this system in the rat? What concentrations might we achieve in the olfactory bulb? And would those concentrations be sufficient, or be in the range that we might think about neurotrophic factors, proteins, peptides, that they could be active if they were very potent? And the answer to that was yes, we could get actually nanomolar concentrations in the olfactory bulb of rodents. And I don't really have time to get into it, but the different tracers we used sort of singled out intracellular axonal transport. That's with the WGAHRP, which is internalized into olfactory sensory neurons and then transported slowly into the brain. And then the HRP itself, the horseradish peroxidase, which doesn't have that ability to get into the neuron, which actually took an extracellular pathway. And the concentration was a lot lower, and I didn't know it at the time, but ultimately we really focused on the extracellular pathway as opposed to the intracellular pathway. And so this work and some of the other work in the field and some historical work really gave rise to this initial idea of an olfactory pathway, namely that you could deliver drugs and that components of the olfactory neurons in the olfactory mucosa could allow for a direct pathway bypassing the blood-brain barrier. So not absorption into the bloodstream, not absorption into lymphatics, but a direct path. And since that time, there's been a lot of work that's been done and with various tracers. So this is, again, a cross-section of the olfactory bulb with the wheat germ agglutinin and horseradish peroxidase. And you can see in blue is the distribution, and you can see it on the surface of the olfactory bulb. Down here is a fluorescein, a fluorophore labeled dextran. And so here's the nasal cavity over here in the rodent. And then you can see here we are in the brain. So there's a cribriform plate. There's a decalcification that's been done here to do the imaging. But you can basically see there's a lot of green here in the nasal cavity. And then there's also green over here on the olfactory bulb side. So the olfactory nerve layer, the glomeruli, the synaptic points between the olfactory neurons and the mitral and tufted cells over here, and then this external plexiform layer. So there's a lot of data that suggests that there is a path into the brain. And I would say that, like I was using modern methods, modern back then, in the early 90s to show this, but, there's long been an interest in this potential of a pathway, and it goes back 100 years. And so, and even more importantly, and I just want to mention it because it was important in the research that I did, an even longer history of looking at these pathways in the reverse direction. So injecting substances, tracers, viruses into the brain or into the cerebrospinal fluid, and then looking at the drainage out of the brain and some of that drainage implicating these olfactory pathways. And I say that this is important because I read all that literature as a graduate student, and that informed how I thought about and how I studied and how I proposed the pathways going into the brain. So a little more historical work in the early 80s, it's worth mentioning the late Helen Sayre at Brown University, Michael Bradbury, who I met a number of times, King's College, also late deceased. But they did some beautiful work. And this is all looking at clearance out of the brain, but, identifying these pathways. And I think they're really, I want to single Helen Sayre and Michael Bradbury and Brad Westrup out specifically, because if you look at this schematic, this diagram, and I would encourage you to go back and look at the original source literature on this. I'm just showing you the schematic because it's easier to understand. It's pretty much got a lot of the things that I ultimately leaned on later on in describing the distribution, what I showed you before. Olfactory pathways, trigeminal is going to come in, and then perivascular distribution. So this perivascular space in the frontal lobe. So they're kind of putting it all together here. Beautiful descriptive work, mostly with radio-labeled molecules. And the idea that the lymphatic system may play this important, the aspect of lymphatic drainage across the cribriform plate into the nasal mucosa may be playing an important role in the drainage of CSF. This was really the motivation for it. When I was a graduate student, I came across this paper by Brinker, an active neuropath, that was really nice, and it was using X-ray contrast medium and in a rodent, and so something called isovist. And so, studies like this is all the old historical work that was there when I was starting out in the field. And you can very clearly see this in the rodent here, that this drain. So here's the olfactory bulb, here's the hemisphere of the brain. And then basically this is this area where you very clearly see the contrast going into the olfactory mucosa. So I mention this just because now if we cut over to today, there are some beautiful modern techniques. And one, I think, of the best ones is to use, where maybe some of you are aware of this, where you can optically clear organs or even the whole head or even the whole mouse. I do this at Denali. And then use a light sheet microscope to visualize in 3 dimensions the distribution of substances. And so this is one such experiment using this Miltenyi Biotech Ultra Microscope Blaze. This is a microscope that I've acquired here at Denali. And so this is using intracisternal injection of this labeled ovalbumin, so a fluorophore-labeled ovalbumin, a 43 kilodalton protein. So it's infused, injected over here, and then we're looking at the brain of the rodent. So this is rostral and this is caudal. And so what you see over here is this distribution. There's a perivascular access. So this is the middle cerebral artery of the mouse over here. And there's a lot of ventral distribution of tracer, and it goes all the way forward to the olfactory bulb and the cribriform plate. So now if we move the field of view forward, this is what this is down below, and adjust the settings to look at this drainage. Now this is ovalbumin around the olfactory bulb, draining across the cribriform plate into the olfactory mucosa. So this is sort of the modern version of what I showed you a moment ago with X-ray contrast, but you can potentially have single cell resolution here. So these kinds of studies are now informing much more on these pathways. And before leaving this topic, I just want to say that, there's also a lot of really beautiful, I think a lot of you are clinicians, MRI, advanced MRI imaging using different methods. I really like Moni DeLeon, my former institution at New York University, published a paper in 2017. And I don't have time to really get into the details with any of this, but basically using these various different methods provided some evidence of olfactory drainage in human beings. And so it's here. You can see that here as well with an intrathecal gadobutyral contrast. And then over here again with another intrathecal contrast experiment showing this very clear olfactory drainage. So I can't really emphasize enough how important that historical work and this, studying the drainage out informs what might go in, because basically the idea is these pathways could potentially be bidirectional. And again, I'll show you a historical drawing over here from Helen Serre I mentioned before. And it relates to some of the topic that we're going to get into, because I know that Jeff is interested in the trigeminal nerve. And so this old, 1992 drawing, which I was, of course, reading and, absorbing all this literature, not only do you have this idea of olfactory drainage, but I'll just point out that, you got the optic nerve, you've got the trigeminal nerve, vestibulocochlear. There's some of what we're going to hear today in the talks, but the trigeminal nerve specifically. And so even, today it's even controversial a little bit whether, these drainage pathways really exist. Yoni Kipnis at Washington University, with his focus on dorsal meningeal lymphatics, has called that into question, which I think is it's the wrong idea. He's totally wrong about that. There's no way you can stop this drainage. If you do an experiment, you're always going to see it. So this is with an intracisterna magna application from my lab. This is published work at the University of Wisconsin, where you very clearly see this beautiful drainage to the olfactory mucosa. And I told you I was going to show you the scrolling pattern of the turbinates in the rat. So green is where we have the tracer that was given intracisterna magna, and you very nicely see that over here in the lamina propria and the submucosa. And I love the beauty of the turbinates there. Okay, so now let me switch gears a little bit. So this work that I was doing in the late 90s, about 25 years ago, knowing nasal anatomy and specifically knowing cranial nerve anatomy, and reading some of that old Helen Sayre work, I had the idea that potentially maybe other nerve components might be facilitating some aspect of delivery from the nasal passage that I was studying. And this is really the genesis of the trigeminal pathway, which a lot of people will attribute to my discovery. And so it was in this paper 20 years ago, but it was work that was initiated about 25 years ago. And so the basic paradigm that Bill Fry and I hit upon in at the University of Minnesota was to do this experiment with I-125 radio-labeled tracers. And the basic idea was to give a molecule intranasally. So this is I-125 labeled insulin-like growth factor 1. It's about an 8 kilodalton protein. And you measure this blood versus concentration time profile. And we were looking at acute delivery, so about a half an hour, and then you sacrifice the animal, you take the brain out, and you can do autoradiography and all these different things. But the basic paradigm was then to match that with an intravenous dose in different animals so that we could try to disentangle or deconvolute what's the direct delivery to the brain and what is maybe going to be accomplished by absorption into the bloodstream. So when we look then at the brain levels, whatever we're seeing in the brain with the intranasal approach that's above what we're seeing with intravenous, we could ascribe to direct brain delivery. And so what you can see here is we have 2 to 3 orders of magnitude higher concentrations in these rats with intranasal administration of I-125 labeled IGF-1 than what we had with this intravenous application, this matched blood exposure. Okay, so that's the first, the paradigm is important. But one of the things that bothered us or bothered me was that anatomically, the distribution in the brain with intranasal didn't make complete sense given what we knew at the time. High olfactory concentrations and then lower concentrations in other brain regions made total sense if we're coming in to the olfactory bulb and then distributing from there. But this peak in the hindbrain didn't make sense. So how to explain that? That was question number one. And then the other explanation was this is 30 minutes. How do we get this rapid distribution to other structures? So those were the two questions that I really concerned myself with. And so this shows you some autoradiography from those early days. And so we used high specific activity solutions, so no cold in the administration solution, to emphasize where we distributed into the brain, and suffice it to say that we went to areas where a high level of IGF-1 receptor was, so that we're really looking at sequestration. But I also did another paradigm where I had a lot of excess cold. So I-125 labeled plus cold. And there we're competing off the specific binding, but revealing a little bit more of the entry. And if you look over here, this autoradiography parallels sort of, goes along with what I just showed you. There looks to be a spike up here at the rostral brain at the olfactory bulb, but there's also signal back here in the hindbrain. And you all know, the hindbrain, pons. So then I started to think, well, what nerve might carry us back there? And obviously it's the fifth cranial nerve, trigeminal nerve. And so when I really started to look in on that, then I discovered that some of the highest concentrations that I could measure in the brain were actually in the trigeminal nerve and not the olfactory bulb, but really the trigeminal nerve. And so this work was published in 2004, and it basically was the genesis of the trigeminal pathway. There's a lot more that I can show you here. So then that second question of how do you distribute once you're in the brain to widespread areas? And that involves these perivascular spaces. So this is some nice work using Texas Red labeled dextran tracers by my former postdoc, Jeff Lockhead, a number of years ago. And so here you see, so this is the whole brain, the olfactory bulb of the rat. And then you see back here, this is the hindbrain. This is the trigeminal nerve root entry zone, But what I want to focus you on, I'm going to actually zoom in so you can really see it, is this. So here's the circle of Willis. So you have the anterior circulation, the middle cerebral artery, the anterior cerebral artery, and then back here is the posterior circulation. So the vertebral arteries, the basilar, and what you should notice, so this is intranasal administration, and we perfuse the animals. We've exsanguinated them. So you're not looking at any luminal, this is not signal in the blood vessel. This is signal in the wall of the blood vessel, the perivascular space. And so it's this perivascular compartment then that is speculated, that I speculated that back then actually accounts for the distribution from the olfactory bulbs and the brainstem, the hindbrain, to then these further sites from there. And that perivascular transport can go fast, can go based on the kinetics of essentially a bulk flow process in that perivascular space. There's a lot of physiology that I'm glossing over here, but I've studied all aspects of that. So, a summary of the rodent work. So we have the rodent nasal passage. A lot of the earlier work just took the brain out and tried to quantify the levels in the total brain, which misses all of this important anatomical detail that I just mentioned. And so in order to really understand this, you have to put it all together. You have to have the olfactory nerves, you have to have the perivascular, take into account the vascular architecture of the brain and the olfactory bulb and the hindbrain. And then certainly the trigeminal nerve. And so this is really work that I think I'm still known for today in the field of isolating out these different anatomical pathways using, as I showed, the old historical radio tracer work, and now modern, is really using a lot of fluorescence microscopy. So we, in the past, really carefully studied the distribution across the olfactory epithelium, the ultimate distribution into the olfactory bulb, the subsequent distribution in the brain along perivascular spaces. That basically led to a lot of hypothesis that then has been tested in the human brain. And maybe just for a couple of minutes, the key question for the field, and Joe, I think you mentioned it before, so then the species differences, how does all this work in the rodent? So I'm very confident, I'm completely confident that these olfactory and trigeminal pathways are there in the rodent. Now, if you're thinking about translating that to the human being, this is where I am today. I've been at Denali Therapeutics for 6 years. Translation is where, the space I am in. Then the species differences are really important to consider. So, what's the evidence that any of this would work in a larger brain, larger distances? So, again, there's some work that I'm known for. I think some of the only really detailed quantitative work in the macaque. So this is the cynomolgus monkey 2008 publication, and then some unpublished work. And where we were administering I-125 labeled about a 20 kilodalton protein. This is beta interferon 1B. It's a therapy for relapsing and remitting multiple sclerosis. And so we were trying to see if we could deliver this into the brain directly. And the long story short is looking at some of these same pathways with the same techniques we applied in the rodent. So doing a really thorough necropsy on these cynos that I did, we can basically show that the olfactory bulb and trigeminal nerve are also these hotspots in the cyno, in the macaque. And so there's a smaller percentage of the dose that reaches the brain, and so that suggests that these larger distances are playing a role, but at least the pathways are operational. And so that was very important. There's a lot of work that I haven't been associated with that has shown over the years, hinted at intranasal administration of mostly peptides can have activity in the brain. Jan Born and colleagues over 20 years ago published a real key paper in Nature Neuroscience looking at MSH and then also insulin, and basically doing lumbar puncture and showed evidence that these peptides were reaching the cerebrospinal fluid. Then Michael Kosfeld published a paper in Nature, which was really important. There's a lot of work on oxytocin. So I'd say, the big, you mentioned, Joe, I think the examples of intranasal, recently, so there's the epinephrine example, naloxone. I'd say calcitonin for postmenopausal osteoporosis. Those are kind of the go-tos, I would say, for my examples, if I had given a longer talk and focused on that. The example for brain delivery has got to be oxytocin. So if you go and there's a rich literature there showing a pharmacodynamic response. And so, the big question still, though, is translating all of this successfully into the clinic because it hasn't been successfully translated. There's not an FDA approval of intranasal oxytocin yet, or intranasal insulin for that matter. So, my conclusions are that, and Joe mentioned advantages and disadvantages of the nasal route, so I won't go over that. We know a lot from the rodent work that I mentioned before, the steps getting into the brain, olfactory and trigeminal pathways, subsequent distribution along the perivascular spaces. Really, the huge question is the translatability to humans. We know a lot more about the physiology, and I've contributed, I think, over the years, I'm proud to say, and a number of papers on that. And then small molecules I didn't mention before. I think, Joe, you also mentioned this, so chemotherapeutics, oncology, parallel alcohol, rich history of intranasal parallel alcohol, including in human beings in glioblastoma. So some of my work, you can go and look it up yourself, but trying to understand the direct pathways. Is there really an intranasal delivery advantage for the small molecules? So yes, that's it. And if